Good morning, students. Today we'll study about ovary, the structure of ovary, internal structure, and the process involved in the release of female gamete, which is the ova, and female formation of female gamete, which is necessary for the process of reproduction. So let's start with the diagram first, and then the process involved. So here you can see this diagram. This is the DS of ovary transfer section. These layers are the single cell structures, show the germinal epithelium, then the primary follicle, layers of follicle with primary oocyte. This is the secondary follicle with layers and the primary oocyte. Then again the large cavity inside the follicle. Here secondary oocyte is released with corona, and secondary oocyte is having zona pellucida or corona radiata, and the process is termed as ovulation. This is the corpus luteum. Then the corpus luteum, which is having a large cavity, a mature graphene follicle here, and this is the degenerating corpus luteum. Now about the structure first, about the process. In the process, the germinal epithelium, these germinal epithelium, they start dividing mitotically. After mitotic division, the oogonias are formed, and these oogonia, they start moving towards the center. After mitotic division, the oogonia, they start moving towards the center. When they start moving toward the center, the one which is larger in size forms the primary oocyte. One which is larger in size forms the primary oocyte. The rest cells, they form follicular cells. Follicular cells, they surround the primary oocyte, and the follicular cells are also termed as the granulosa cells. So the rest form follicular cells or granulosa cells. Larger cells, they form primary oocyte. Primary follicle, the number of primary follicles keeps on increasing by the division, mitotic division. The layers increases in number. When the layers increases in number, a new theca is formed. Theca is covering. In this new theca is formed. Follicular fluid is added, and more layers are formed. New theca is added, and the fluid is added. After this, it becomes a secondary oocyte, secondary follicle, and primary oocyte. After the meiotic division, forms the secondary oocyte, which is present inside the secondary follicle. Once again, germinal epithelium after mitotic division start moving towards the center. The larger ones are termed as primary oocyte. They are surrounded by the rest cells. They form the surrounding cells or the follicle cells or the granulosa cells. The number of granulosa, the number of follicles, it increases, and they form the secondary follicle. Layers are added. New theca, which is a covering, new theca or cavity. A cavity is formed. Follicular fluid is added. After this, and the primary oocyte, which is present in the center, it gets divided mitotically to form the secondary oocyte. So two stages at a time. Follicular cells are added, cavity is formed, follicular fluid is added, and this primary oocyte gets divided meiotically to form secondary oocyte. This is about the secondary follicle. Then again follicles are added, and the cells of the follicular cells, they merge together to form a large cavity. The follicular cells merge together to form a large cavity, and this large cavity is termed as antrum. Antrum, it is a fluid, follicular fluid is present inside this large cavity, and when it gets measured, then few more layers are added. Follicle cells, after merging, the follicle cells grow in size, and when they grow in size, they are the follicular cells, they rupture, and releasing the secondary oocyte. Secondary oocyte is covered with a layer termed as corona radiata or zona pellucida. So this, and if it is not released, then in the corpus luteum, the mature follicle, which is termed as corpus luteum, it degenerates. If the during ovulation it is not released or not formed, then it degenerates. So this is about the structure of ovary. Again, this I am repeating. Here germinal cells after mitotic division move towards the center. Germinal, the larger ones form the primary oocyte. The smaller cells or the rest cells, they form the primary follicle. Primary oocyte undergoes meiotic division, forms secondary oocyte, and the follicle layers of follicle cells are added. Layers of follicular cells are added. Theca is added. Follicular fluid is added. In the next step, the follicle cells, they merge together, they form a large cavity termed as antrum. The cavity is filled with a fluid called the follicular fluid with the antrum, and the secondary oocyte is present. Here only secondary oocyte, the same follicle, it increases in size. The cavity also increases, the follicular fluid increases, and a mature follicle, which is a graphene follicle, which contains a larger cavity termed as antrum, it ruptures. The size of the graphene follicle is about 1 centimeter in size, and it ruptures, releasing the ovules. It ruptures by the process of ovulation, releasing the secondary oocyte. Secondary oocyte is covered with a layer termed as corona radiata or zona pellucida. The rest part, secondary oocyte is released. The rest part, it forms the corpus luteum. That is, it may be asked to define what is corpus luteum. So after releasing the secondary oocyte, the graphene follicle is termed as corpus luteum. After releasing the secondary oocyte during ovulation, the graphene follicle, this is the graphene follicle. Graphene follicle. So after releasing the secondary oocyte, graphene follicle is termed as the corpus luteum, and which degenerates later on. So this is all about the structure. Just learn it properly. Try to first understand the steps involved and learn the diagram. Diagram is TS of testis, TS of ovary, and the process of formation is very important. These two are very important questions for examination point of view. Thank you, students.